Today we're going to start section 2, which is our introduction to genetics. So to begin, I want to start by asking you a couple questions to think about throughout this unit. How are traits passed from one generation to the next? Why do offspring sometimes look different from their parents? How can traits skip generations? Can we predict the outcome of genetic crosses? So these are all some questions to keep in mind throughout our section on genetics and answers that we'll be able to answer as we cover our genetics unit. So to begin, I want to start with just some vocab that you need to know. So do write this down in your notes. So the first word to cover is genetics. Genetics is the study of genes, heredity, and variation in living organisms. Next, a gene is a unit of heredity in a living organism. It codes for a protein or RNA chain that has a function in the organism. Heredity and inheritance is the passing of traits to offspring. And a trait is a characteristic that can be passed from parent to offspring. So make sure you copy these vocab words in your notes. We will use them throughout the entire genetics chapter. So it is important that you know these words and you know what they mean. So for centuries, we have known that traits are passed from parents to offspring. Duh. I mean, of course we know. There are families who all have the same color of hair, or they all have maybe like the same nose or chin structure. But what was not always understood was how those traits are determined. So one hypothesis that was very popular for centuries was that the traits of parents were blended or mixed in offspring. So I want you to think about this for a second. What evidence can you give to support this hypothesis? And what evidence can you give to refute this hypothesis? All right, so hopefully you've thought of some examples that would support the hypothesis that traits from parents are blended, and hopefully you've thought of an example that would refute that hypothesis. So one I can think of would be height. So let's say you have a really tall parent and a really short parent. If it was blended, you would expect the offspring to be medium height. In this example, if we were going to refute the hypothesis, let's say we had an average size like father and a short mother and a really tall offspring, taller than the father, that would refute that hypothesis. Again, let's maybe pick on hair color. Let's say one of the parents had really dark hair, the other parent had really light hair. If traits were blended, you would expect a medium hair color in the offspring. If this hypothesis was going to be disproved, then you would have maybe a medium-haired parent and a light-haired parent, and you'd have like a dark-haired offspring. So genetics really begins with a man named Gregor Mendel. He was an Austrian monk, and he began studying inheritance in the 1860s. So he focused on pea plants, and he really studied how traits were passed from one generation to the next. So why pea plants? So pea plants were a good plant to study because of the structure of the pea flowers. There's a presence of very distinctive traits. There's a fast reproductive cycle, and it was easy to grow hundreds of plants. So the structure of the pea flower allowed Mendel to isolate important variable in his experiments, which is fertilization. Pea plants can self-fertilize. So in this process, the plant's own pollen fertilizes the pistil on the same flower. That produces a purebred plant. And pea plants can also pollinate other pea plants in a process called cross-fertilization. And that produces a hybrid plant, which is a plant with different forms of a genetic trait from each parent. So the structure of the pea flower makes it relatively easy to control whether the plant self-fertilizes or cross-fertilizes. So as Mendel studied pea plants, he was able to figure things out that others hadn't been able to discover. And the reason why is mathematics. So up until the 1860s, most biologists didn't consider mathematics or probability when they studied living things. And so Mendel, he recognized that if he made enough observations, he could use statistics to test the blending hypothesis. So in order to test the blending hypothesis, Mendel used different pea plants. He saw that some pea plants produced yellow peas and others produced green peas. And so in order to test the blending hypothesis, he had to predict if you crossed a yellow pea with a green pea, what color would result if they blend. And so if the blending hypothesis were true, Mendel should have observed a chartreuse pea. Okay, so this color right here, so the mix between yellow and green. However, Mendel performed thousands of cross fertilizations between yellow and green peas, and he never observed a blending of a pea color. Instead, the peas were always either yellow or green, but never both. So why do you think it was necessary to observe so many genetic crosses? Why couldn't you just do it with 1 or 2 pea plants? Hopefully you're thinking about somewhere along the lines of like all the different possibilities that 2 parents could make in an offspring. So think about like you, for example, especially if you have a lot of siblings, that your parents could make a lot of offspring that look different. So he wanted to have lots of trials to make sure that there is no way that there could be a blend between yellow and green. So as Mendel continued to cross-pollinate yellow and green peas, he started to discover some patterns. Okay, so let's take a look at the picture up here. Okay, so here we have the parent generation. So he has 1 yellow pea parent, and he's going to cross it with 1 green pea parent. Okay. Next is the F1 generation. That stands for the first filial generation, or you can just think like their first set of offspring. Okay. He noticed that all of these were yellow, which was surprising. So that tells him that the yellow pea color had to be dominant over green. It just had to. That's why all of the F1 is yellow. Then he let those self-pollinate, and he noticed that in the F2 generation, or the first generation's offspring, he noticed that some of them were yellow, but some green peas also appeared. And after so many crosses, he was able to pick up on a trend. He noticed that for every 3 yellow peas, there was 1 green pea. So they formed in a 3-to-1 ratio. All right, so here's the same picture. It's just showing the offspring of all the peas. So go ahead and copy this one down in your notes. Make sure you label the generations. So at the top, we have the parent generation, where we have a yellow pea being crossed with a green pea. Their offspring resulted in 4 yellow peas. Okay, so again, that's our first one, or our F1. Then we can show the offspring if the plants were allowed to self-pollinate. Okay, so here's the F2 generation coming from this yellow pea. So the offspring of this yellow pea was in the 3-to-1 ratio. So 3 yellows for every 1 green. Then these were allowed to self-pollinate as well. And he saw, so this yellow produced 4 yellow offspring. This one produced them in a 3-to-1 ratio. So 3 yellows for 1 green. This third yellow produced offspring in a 3-to-1 ratio, 3 yellows to 1 green. And then the green pea plant, when it self-fertilized, produced all 4 green pea offspring. So what does it mean? So Mendel reasoned that the only way to explain the numbers he observed was that the offspring must inherit 2 alleles for each single trait, 1 from the mom and 1 from the dad. He also came to the conclusion that 1 allele was dominant over the other, but the other was still present because it could be passed on to the next generation. So in the pea plant example, the yellow allele was dominant, but the green one was still there because every now and then there'd be a green pea offspring. So in addition to seed color, Mendel observed 7 different traits in his pea plants. So he studied seed shape, he studied color, flower color, pod shape, pod color, flower position, and stem height. And all of these 7 traits followed the same rules that he observed with the pea color. Before we continue with Mendel, I want to take a pause and just go over some vocab words. So some of these are words that I talked about in the past couple slides, and some are going to be new vocab words that I'm going to talk about in the next few slides. So the first word I want to talk about is an allele. So an allele is a distinct form of a gene. So if we go back to the pea pod example, that would either be the yellow or green. Next, we have dominant allele. So this is the form of the gene that is fully expressed when 2 different alleles are present. So going back to the pea pod example, yellow is the dominant allele because that's the color that we saw when we mixed yellow and green. And so dominant alleles are always given the symbol in a capital letter. Okay, so since it's yellow, we're going to give it a capital Y. Next is the recessive allele. This is the form of a gene that is not expressed when paired with a dominant allele. So in the pea pod example, this is the color green. And instead of giving it a G for green, it actually gets a lowercase y. So since we're looking at color, they do need to have the same letter because we're looking at the same gene. So then capital letter corresponds to the dominant allele, the lowercase letter corresponds to the recessive allele. Next is a genotype. The genotype is the genetic makeup of an organism. And I remember this one by geno. So by geno, think genes. So what are the genes of this organism? So there are 2 options when we looked at that F1 generation. So the first option could be you're getting the yellow gene from each the mom and the dad. So your genotype would be YY. Or you get a yellow gene from the mom or a green gene from the dad, and your genotype would be capital Y, lowercase y. Next is the phenotype. The phenotype is the outward expression of the trait, and I remember this is by what you, like the physical trait. So I remember phenotype, physical, so what you see. Okay. So if we go back to this example, our 2 genotypes could be YY or capital Y, lowercase y. Because they have the capital or the dominant, they're going to be yellow. Okay. So the genotype is the genes. The phenotype is what you see. So I would see yellow peas. And then finally, since we're going to look at organisms with 2 alleles, we have words to describe whether or not the alleles are same or different. So 2 same alleles are going to be called homozygous. Okay. So homo means same. So 2 same alleles. So that could be capital Y, capital Y. Or in the case of the green peas, it could be lowercase y, lowercase y. Okay. So homozygous means same. And on that note, I don't have heterozygous on here, but let's write it. So another term we can use is heterozygous. Hetero means different. So 2 different alleles. So for a heterozygous pair, that would be a capital Y and a lowercase y. Okay, they're different. We have the yellow and the green. All right, so let's come back to Mendel. So Mendel's work provided some key insights into genetics. So some things we learned from Mendel are traits are inherited in certain numerical ratios. We learned that heredity factors do not combine, but they are passed intact to the next generation. So that means like there's no blending. Each member of the parental generation transmits only half of its hereditary factors to each offspring. Certain traits are dominant over other traits, like yellow is dominant over green. And different offspring of the same parent receive different sets of hereditary factors, which means that they receive different genes. Right? Some of their offspring, in the F1, they could be capital Y, or they could have been capital Y, lowercase y. Okay, so they get different sets of those hereditary factors. So Mendel summarized his findings into 2 laws. His first law is the law of segregation, which says when an individual produces gametes, the copies of a gene separate so that each gamete receives only one copy. Another way to word this is 2 alleles for each trait separate during meiosis, and then during fertilization, 2 alleles for that trait are going to unite. His other law is called the law of independent assortment. Alleles of different genes sort independently of one another during gamete formation. So an example of this would mean there's no correlation between an elephant's trunk length and its tail length. So whatever genes control the trunk length are separate from the genes that control the tail length. Okay. So this is only true for genes that are not linked together. Later on at the end of this unit, we will talk about linked genes, and that'll change the law of independent assortment a little bit.